Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we will begin the question and answer section, and I will ask our speakers your questions. Your questions in the question and answer window are only visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speaker, Celal Dari and Zoe Hughes-Thomas from GSK.
Thank you. So thank you, BioProcess International, TCAN and VelitaCell for this opportunity to present. So I'm just going to give a very brief introduction to automating medicine design within GSK before I hand over to Celal Dari, who is a second-year apprentice within my automation team, who will talk about the more interesting science and data. So within GSK, medicine design organization has accountability for designing and delivering our drugs, and that includes both the small chemical molecules and the large molecules, so the biopharmaceuticals. Within medicine design, the medicine design automation team has the accountability to drive the automation strategy across medicine design. And one of our key focus areas is to use automation to accelerate the cycle times and increase our success rates from target to candidate molecule. On this slide, I've just got a snapshot of some of the automation that we've used, and I'm sure many of you will recognize many of these pieces of equipment. So we use automation across sample management, medicinal chemistry, high-throughput screening, developability screening, protein expression, and phenotypic screening. So the role of medicine design automation, there's really three core objectives. One of which is that we support the scientists within medicine design if they are bringing in large integrated automation solutions. So these are ones that are fully integrated end-to-end workflows and generally require specialist training for people to use it and are usually run by dedicated groups. We also bring in small modular solutions. So automated work cells, which have open access, so a kind of reader feeder type where you will have a hotel feeding and piece of instrumentation. And one of the key parts of our group actually is working with an engineering team who are embedded within our team to provide that first line troubleshooting and rapid support. And one of the key pieces of medicine design automation is to maximize that value of the automation we have and therefore minimizing the downtime. We've also created a network across the whole of the community within GSK where we can link together automation savvy scientists and engineers so that we can share best practice and learning. And one of the key pieces of this is that we work with the scientists to embed new technologies into GSK. And this is a segue into Celal's talk where he'll talk about how he's used a new technology to reduce a QC step within our protein expression platform so that he can high throughput quantification of IgG.
Hi everyone. Today we're going to introduce you to the Titer IgG assay, which I have been working for the last year. So you may ask, what is Titer? Well, Titer is a 96 or 384 high throughput antibody quantification assay, and it's based off a FIT-C probe conjugated to protein G. It exists in two concentration ranges, including 2.5 mg per liter to 100 mg per liter and 100 mg per liter to 1,500 mg per liter. That was a mouthful. So the biggest USP of the assay is its quick preparation time that literally anyone can do, even Boris Johnson. Our own preferred protocol includes using 50 microliters of media or PBS and using 50 microliters of sample. The plate is loaded onto the TCAN Spark Cyto and a readout is produced. The results are easily derived by creating standard curves on the Velita app, which you can get for free with a box of the plates, or you may use the standard Microsoft Excel. We've demonstrated a strong correlation with existing IgG quantification methods. And when comparing the liter titer to octet, which is what we use in-house in GSK, the liter titer presents the advantages of being a much quicker assay with very few steps and using a multi-mode reader, which can be used for other purposes like cell imaging. This obviously improves workflows and allows the time from molecule to medicine to be reduced, which further supports our pipeline. And I'm sure many of you here will be joining from pharmaceutical companies and will be interested in this. So just a bit about the mechanism of the assay. So as previously mentioned, the plates are coated with a FIT-C probe, which is conjugated with protein G. The protein G binds to IgG antibodies. The unbound probes rotate faster, the bound probes rotate slower. The Spark cyto can differentiate the rotation and produces a fluorescence polarization readout, with the units being the Miller polarization. And these units can be plugged into a standard curve and a result can be interpolated. So, and we've done some improvement in our workflows with the liter titer, using it on the TCAN Spark, and we observed that incubating the plates inside the Spark cyto actually improved the protein G binding, which is most likely due to increased kinetic energy. In addition to this, the probe binding reaches stability around 5 to 45 minutes. So we've discovered that the assay must be used in less than an hour, or the probes start to lose binding ability. And to improve our workflows, we've also pre-prepared IgG standards to actually make it easier for scientists to run this assay. So we've conducted experiments using the liter titer where we measured the stability of our IgG standards diluted in 2 different types of Chinese hamster ovary media, and we observed that there wasn't any difference over the 4 weeks to the MP signal. IgG standards were prepared using a TCAN fluent by another member of our team. Duplicates of the standards were created, one in CHO production media and one in CHO selection media. And we stored this at 4 degrees Celsius in a cold room and took readouts once a week for 4 weeks. We observed that there was no major difference in MP signal between CHO selection and CHO production media. We demonstrated a good correlation between MP signal and IgG concentration, and there was a significant reduction in titer analysis turnover times, where a request for octet reading in GSK could take a week in our department, whereas this assay was less than 15 minutes and can be done by scientists themselves, which meant that we were able to obtain more data with a short amount of time. So in terms of how this assay can benefit your work, this assay can be used to evaluate the effectiveness of integration. It can be used for predicting expression and supporting purification tuning. There is also potential to a fully integrated inline analytical feedback for automation, which could really innovate R&D. And this slide sort of shows how we picture the future of automated IgG analysis to be. Ideally, there would be minimal human intervention, and the throughput could be as high as running 384 samples per 15 minutes, which can support our biopharm pipeline. And lastly, to close it off, I'd like to acknowledge these people on the list, as well as the host BioProcess International, for hosting this webinar for us. And I open the floor up to any questions, should anyone have any.
Great. And just to note, joining us for the questions are experts from ValitaCell and TCAN, in case anyone has any technical questions. So the first question, and go ahead and type your questions into the chat window. The first question is, do I need to remove cells from the sample or purify the protein before running a Valita titer?
So no, unlike other IgG quantification screening tools, the liter titer is robust to cellular contamination up to 15 million cells per mil. This means that there is no sample prep, pre-purification or centrifugation to remove cells required prior to analysis. Samples can be analyzed neat straight from culture to the liter titer plates once they fall within the functional range of the assay.
Okay. And how many reagents does the liter titer require?
So the liter titer does not require any additional reagents or buffer. The plates are pre-coated with a Velita titer probe based on a FITC labeled FC-specific binding peptide. And the binding peptide is a derivative of protein G binding domain 1, aka GB1. So you will only need the Velita titer plates, your cell culture media, your IgG standards, and your IgG samples to be quantified.
Okay. Well, thank you to our speakers, Zoe and Celal. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Let me see. I did just get 2 more questions in. Are you still there, speakers?
Yep.
Okay, let's go ahead and do these. So how many dilutions per sample do you use? Meaning, do you assess dilutional linearity for reportable results?
So I think it really depends on what the concentration that you have and what purpose you're doing it. And if you're really needing an accurate result, then yes, we would do dilution split for linearity. And if you're looking for a ballpark figure, to understand if you have merely got expression and what kind of range you're looking for, then we would start with, it's having an understanding of what level you might get. You just start with whatever dilution that is and run that across the plate. I think the other thing really is the throughput that you're doing it at, and therefore how many dilutions and how many samples you can run at once.
Okay. Do you know if this assay can work with bispecific antibodies or FC fusion proteins?
I'd like to pass that question to Velita Cell, as they'll be more equipped to answering this question.
Yeah, absolutely. Thanks, Celal. I'm answering from Velita Cell, and yes, Velita Titer is able to quantify also bispecific and FC fusion. The important thing is those constructs need to have an FC domain. So our probe protein G domain GB1 is able to bind to any FC domain. Depending from the molecule, we might need to optimize a little bit the workflow, but yeah, in principle, it's absolutely able to quantify both bispecific and FC fusion.
Okay, and have you performed a cross-validation using a LC?
We haven't ourselves, but I can again pass this off to Velita Cell if they've done any cross-validation.
I believe he means to HPLC. So yes, we have a peer review publication where we have compared the performance result of Velita Titer with HPLC. So that's an available peer review publication available.
Okay. And when you use neat cell culture samples, what matrix do you prepare your standard curve in?
So we would use any IgG standards prepared in the media that your actual real-life samples are in. So if it's CHO production media, we would say put your IgG standards in the media because there's obviously background fluorescence.
Okay, great. So I think we have answered the additional questions. Thanks for getting those in. And we look forward to having you join us at future webcasts. Thanks again to our speakers and to you, our audience, for joining us. Goodbye.
Thanks. Goodbye.
Thanks everyone for attending.